evening, all of you.
Good evening, ma'am.
Are you done?
Yes, ma'am.
Good evening, all of you. Why all of you are packed this side and this bench is all empty? Why, Aisa? What you? Come here in the first bench. Some of you come here. खींचना पहले जाके पीछे बैठती है, फिर उसको सामने खींचना पड़ता है, ऐसा क्यों? नहीं, I won't sit that. I sit on the outside. What did we learn yesterday? Girls, come up here. सामने आ जाओ. यहाँ पे भी बैठ सकते हो. One of you can sit here also. हम transpiration. तन्मय चौधरी कौन सा टॉलेस्ट इन द क्लास? हमारा सरनेम क्या है? You also Maharashtrian, no? You also. 90%. कौन सा 90%? ऐसा कैसे होता है? 90 और 10 का क्या रेशियो होता है? अच्छा, obviously. Were you there yesterday? No, no. You? Yeah. You were there? मतलब कल लिया तो आज नहीं लेना है ऐसा? Yesterday you were not informed. How many of you were not informed? You were not there. Ma'am, no one is supposed to be not informed, ma'am. They have to check that. This also came late. This was there, but this was dozing. This was not there. This was online. You were there. तुम लोग मेरे लिए सामान ही हो बस. You're just coming and sitting down, most of it. Why? Why? Ah, this one also not there, no? हाँ? अब से Friday, Saturday दोनों. इतना बार. Tell them to start that bio and ICSE also. कि bio में ICSE नहीं होता. I'm not saying. तो ICSE में तो होना चाहिए. Yes. Exactly. चलो फटाफट we'll finish today. Transpiration, very easier chapter. Yes. We'll finish it off. Huh? We just started with the experiments only, and now today I'm going to write. पीछे क्यों नहीं जाता? You all are able to. सर्कस है. ये तुम्हारे स्कूल में से सर्कस दिखाना मेडल मिलेगा. Sorry, I can't give you any medal for this. किसका बॉटल है? किसका बॉटल है ये? चलो 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 check out a book. Online. क्यों? Online क्यों? Online क्यों? कल heavy rain था. बारिश थी, heavy rain नहीं था ना. Take out your book. Get ready with your notebooks. We are going to discuss with, what is it? Which? Photosynthesis already over. Photosynthesis, the diagram-based questions we did yesterday, and the objectives I asked you, do it at home as homework. Kuch doubt hai to pucho. Is there any confusion? Did you try solving at home? Yes. No, I know that answer. No. Whatever you say, it's spontaneous. That is the truth. No. So we had already been talking about those experiments. We had already talked about this experiment. Just for those of you who remained absent. This is an experiment that you have to go through. Very important, very common. You get to see in your question paper. Very often questions are there on this. I will give you certain questions on this. So see that you have understood this. Those of you who were there, those of you who are not there, we were talking about that this is a process by which, this is an experimental setup by which we understand that from the leaves, by the process of transpiration, water vapor is released, and now the water vapor gets condensed to form droplets, and that is what you see on the inner surface of the polythene sheet. So this was his experiment. Next is we were talking about this litmus, the cobalt chloride experiment, where we had learned in this experiment also more or less the same setup with the bell jar that we do, but we only keep an indicator, the cobalt chloride paper, which makes us clear about the presence of the moisture in the environment. And its actual color is blue, and in presence of moisture turns to pink. So that you have to remember, this was the weighing experiment that we were talking about, and some other modifications of the experiment also we discussed. No, just take the pot, initial weight and final weight. Will there be a difference in the weight after a particular period of time? Yes, there will be. Then how do you account for the difference in the weight? We can only say that the difference in the weight is only because of the loss of the water in the form of vapor by the process of transpiration, and hence the final weight is supposed to be lesser than the initial weight. And this is something like a modification again, where we are saying that take a graduated side tube, put a layer of oil. In all these experiments now that you talk about the presence of an oil layer when applied, the presence of the oil layer is always to prevent surface evaporation. No? So that is the standard concept that you have to remember in whichever experiment you come across. And here we understand after a particular period of time, 8 hours, 6 hours, there will be a little bit of drip in the level of the water. And why the usage of the cork? What I said? To stop surface. Prevent surface evaporation again. So these things to remember here. This is again another type of the weighing method experiment where you are taking a shoot in a test tube, 3/4 filled with water. You weigh, take up initial weight, wait for a particular period of time under all normal conditions, environmental conditions, and I'm so worried for my daughter, you know. So now, because I'm mentally getting prepared that she'll behave like this till the 10th standard. So now when we are talking about the spring balance, what is the spring balance used for? I'm here running fast, trying to finish the chapter, and he's smiling away at him. What is this going on? Scary sign of future. The signs are very scary now. So this is what we are talking about, the same experiment. We had stopped with this photometer. Now we were discussing about this. I'll just go through this again for those of you who remained absent. So this is the entire setup in short to understand that you have to take a shoot. The shoot should not have roots and cut the stem in such a way that there should be adequate amount of surface exposed to the water so that the absorption by the stem is as it is slower. So when you cut it on a slant, then when you're just chopping it straight, the surface exposed to water is less. So we slant and cut it so that the surface exposed to water is more and the absorption is a bit faster. So ye hai, and then the shoot should be there. Again, the necessity of putting a cork, you remember why, no? The necessity of putting a cork. Addition to this is a reservoir. What is a reservoir? Source of the water. The main part of the experiment I go to here is the glass tube. Inside the glass tube, a capillary tube. And on the outer side of the capillary tube, the graduated or the calibrated scale. Necessity is we are trying to find out how much of the water intake. Now, for this experiment-based question, which is again quite a common question in your paper, you are asked, what is the significance, what is the necessity of using this photometer? Your answer should be, photometers are devices which can measure the rate of water uptake, intake or uptake. The rate of the water intake, the rate of the water intake. You're not to write the rate of the measure, the rate of the water released. We are not actually measuring this much. We don't have anything to measure this side. We have only to measure this side, how much of the water is taken in. And then what happens? Introduction of the bubble. I already told you yesterday, that's actually a very difficult part of the experiment. And then now that if one bubble is introduced, we have to allow it to go to the 0 mark. We take one reading, allow it to go to the desired mark, maybe now 2. for the first reading. Again, what do we do? How will we bring the bubble back to zero? What did I say yesterday? Open the reservoir, let the water flow back, and then the bubble comes here. And this way to repeat the readings. So after we take a couple of readings, 2-3 readings we take, what are we understanding? What did we understand? We have understood that how much of the water has been lost, how much of the water has been taken in, because that is equal to what is the amount of water lost. Okay, we'll do questions on this. Is this clear? This is very important in this chapter. Clear about the concept of photometer. Next is we talk about types of transpiration. We had been already talking about types of transpiration. Try to understand first of all this. We tried to, yesterday I had already mentioned what are some of the structures necessary for transpiration. Structures which are bringing about the process of transpiration. What are those? One is stomata, and second is lenticels, and third is cuticle. Again, let us run through this first, then we come across here. I talk about stomata. Location kya hai? Lower epidermis. Location of stomata kya likhna hai? Lower epidermis. Location of lenticels kya likhna hai? Bark. On the surface of the bark of the stem, surface of the bark. And location of cuticle kya likhna hai? Upper epidermis. Now, is this a cell? Is this cellular in nature? Yes. Is this cellular in nature? No, it is just an opening. Is this cellular in nature? What it is?
It is a waxy coating.
Fatty tissue, waxy coating, a waxy secretion of the epidermal cells. Stomata, the opening can be regulated by the amount of the water, temperature, all this. So the opening of the stomata can be regulated, and mostly we come across, when is the stomata mostly open? Closed during the night. What about the lenticels? Do they remain open all throughout the day? Yes. Can the opening of the lenticels be regulated by temperature, water, moisture? No. So these are openings which would remain as it is all throughout the season, all throughout the day. What about cuticle? Is it an opening actually that we are talking about? No, it is just a layer. And through the layer also the process of evaporation can continue all throughout the day, but much lesser than the stomata and the lenticels. So now we generally go by this. Whatever amount of the water loss is by the stomata, much lesser is by the lenticels, and even more less is by the way of the cuticle. No, so 3 that we have been talking about. The process of transpiration by stomata, stomatal transpiration. Stomatal transpiration, and as usual we talk about cuticular and lenticular transpiration. 3 types of transpiration that we talk about. When we are talking about cuticular transpiration, can you give me 2 examples to understand that in which type of leaf the cuticular transpiration would be much more, and in which type of leaf the cuticular transpiration will be very less? As it is, cuticular transpiration is much lesser, but even then if I compare, can you give me 2 contrasting leaves? Desert plants. When you talk about the desert plants, the rate of transpiration by the cuticle will be almost nil. Why? Because the cuticular layer in those type of plants is thick or thin? Very thick. So thickness of the cuticle is actually referring to what? Preservation of the water, conservation of the water, not allowing the water to get lost. And when we are talking about the thinner layer, yes, the process of evaporation can continue. Okay. So cuticular transpiration, a concept of transpiration to understand. Next is we are talking about stomatal transpiration. Here, a short process you have to understand. What is happening in the entire process of stomatal transpiration? As we are saying that in the process of transpiration, roughly we generally say that 96 to 97% of the water loss in a plant is by the way of the stomata. But what is the process? The process is elaborate, but just in few steps, let us try to understand. Just in few steps, we try to understand here. Now here, first I'm talking about some few epidermal cells. We are talking about some few epidermal cells followed by some spongy cells, and then if I look into some layers of the spongy cells, we are going to come across कौन क्या हुआ? Who did this great work? किसने किया?
जल्दी बोलो, कैमरा चेक करने के पहले।
सर कैमरा बंद है।
It's not a joke. किसने किया? How come you are testing your strength on that thin sheet of paper? Who is this? You got that thin sheet of paper to test your strength. Tanmay? नहीं किया होगा। तुमने किया? Nobody is honest in this class to confront the truth. A smart person has that much of courage to confront the truth. Yes, I've done it, but by mistake. Sorry, nobody has that courage. Never mind, we will pay for this in the future. Now, I can't waste any more time. Let us go ahead. So now we are talking about. Now, we are talking about the process of stomatal transpiration. Remember the steps. First of all, can you tell me what would be the source of water to the cells? The xylem. So I talk about what is the xylem of stem, leaf.
Yes, leaf.
Xylem of the leaf. No, first of all, I have to understand the xylem of the leaf, a group of cells which will constantly supply water to these cells. Which cells I'm talking about? Palisade cells. So now, palisade, mesophyll. So now, first of all, what are some of the steps to understand? The xylem will, as it is, supply water to the mesophyll cells. Xylem will supply water to the mesophyll cells. Now, this water that is now present in the mesophyll cells, every mesophyll cell, or if I just draw the mesophyll cell something like this, there is a fine gap which is there between these cells, okay? There is a fine gap between the cells, if I have to understand. So this water which is there, the water that is present in the vacuole in each of the cells under a condition of adequate temperature, dry environment, dry and hot environment, adequate temperature I talk about, lack of moisture, lack of humidity if I talk about, a windy day if I talk about, if these are the normal environmental conditions I understand, then what will happen to this water? The first step is an adequate amount of water starts getting evaporated and results in the formation of a thin film of water surrounding every cell. So results in the formation of a thin film of water surrounding every cell. So this thin film of water now, as they collect, when we are talking about, I'm saying that every particular cell will generally have, every particular palisade mesophyll cell will generally have a film of water surrounding. But as I again mentioned that the temperature is adequate, it is a hot and a dry environment mostly that we are talking about right now. And then what will happen to this film of water? The film of water will start undergoing the process of evaporation and result in the formation of water vapor. So the second step is the film of water which starts getting collected surrounding the palisade cells, they start now getting further converted into vapor. And this vapor now will diffuse from one region to another. They will start diffusing from one region to another. So what is this region that I'm talking about? Between the cells present, the intercellular spaces. So intercellular spaces between the spongy cells, majorly, there are enough gaps. So now the film of water, which gets converted into water vapor, will start diffusing. And you know the rule of diffusion, from any concentrated area, it will always try moving towards a low concentrated area. So now the water vapor starts moving, and the water vapor from here it will start moving, and now they start getting concentrated on an area where surrounding there will be a stomata, but this space, a wider space surrounding the stomata where the water vapor gets concentrated, and this space, though it is also an intercellular space, but we majorly call it as a substomatal space. And now that it's a hot and dry environment, again I'm mentioning. So if it is a hot and dry environment, on the outer surface of the epidermal cells, are you expecting an adequate amount of moisture? No, we are expecting a dry environment. So I can also say this way, that the outer environment is having a lower concentration of water vapor. And inside here, there's a higher concentration of water vapor. And that is what is the reason why the water vapor is constantly diffusing and coming out. The water vapor is diffusing and coming out. And this is how the water constantly, or water in the form of vapor, is constantly lost by the leaves. But now the question is that what are some of the factors which is allowing the stomata to remain open? What did you learn earlier? If it is a hot and dry environment, inside the guard cells also which process is going on? Photosynthesis. And this is also allowing the accumulation of water, and accumulation of water is relating to turgidity, and that's how the guard cells are also open. The guard cells are also open. So that's the reason we also say that way, that photosynthesis, that is what is actually happening, is a process good for the plants. Photosynthesis is a process good for the plant. But in the process of photosynthesis, what is the price that the plant has to pay? Loss of water in the form of vapor. So transpiration is also now happening. But yes, though we say that transpiration is the price that the plant pays, but because of the process of transpiration, quite a number of times the plants are saved. Otherwise, they would have an excessive accumulation of the water, which is also not good for the plants. So the process of stomatal transpiration, you have to understand here, and the role of the stomata. What is the role of the stomata in this process? Then next is we come across lenticular transpiration. Only the factors here you have to study, not the process. So when we are talking about lenticular transpiration, when does it happen? All throughout the day. The lenticels openings cannot be regulated. There are no particular factors that we have to talk about regarding the opening of the pore. No, it remains open all throughout the day. But what is the proportion of the water lost by lenticels? Just 1 to 2%. Not more than that, just 1 to 2%, not more than this. So when we are talking about the amount of water lost by the plants. Now, there is one more experiment here that we are going to talk about. There is one more experiment that we are going to talk about. Look at this particular experiment. Which figure it is? 5.9. Figure 5.9. Look into that. What are we trying to understand? Quite related to the stomata now that we are talking about. Okay. What are we understanding here? We take a potted plant. Now here I'm just showing you the leaf. We take a potted plant and select some 1 or 2 leaves like this. And then what do? We attach strips of cobalt chloride and pin them. The cobalt chloride has to be on both the surfaces, on the upper surface also and on the lower surface also. And where should you put the pot? Under sunlight for a particular few hours. After that, what are we expecting is the cobalt chloride. What does it indicate? Presence of moisture. Now, can you tell me the cobalt chloride on the upper surface of the leaf or on the lower surface of the leaf should readily turn pink? Yes, you. What's your name? I forgot. The girl, second last. Anushka, bolo. The cobalt chloride on the upper surface should turn pink faster, or on the lower surface should turn faster? Lower surface. Q. More stomata on the lower surface and more amount of water loss in the form of vapor. So cobalt chloride, the more faster it comes in contact with adequate amount of moisture, it shows a change in the color from blue to pink. Now, this is again, we are talking about adaptations. Now, let us talk about some adaptations. What are adaptations? Adaptations means different environmental conditions which are associated with the rate of transpiration. Different environmental conditions we have to come across. In different environmental conditions, if I talk about a desert plant, if I talk about a plant of the plant very close to the coastal areas, if I talk about a plant on the or a mesophytic plant, normal characteristics like the plants surrounding us now here, will they have the same rate of transpiration? No, they will have variable rates. What is the reason? What are somehow why certain plants have to develop certain characteristics to minimize the water loss? Why certain plants have to develop certain characteristics to maximize the water loss? So that is what we are going to talk about, adaptations. Some of the adaptations, I think 4 or 5 of them are there in your syllabus. Let us just run through them, go through them, some of these. The first is we are talking about here, adaptations. When I look into adaptations, rate of transpiration, we are trying to understand what can be certain adaptations which can minimize the rate. Here we are majorly talking about that, minimizing the rate of transpiration. What can be or minimizing, or in certain cases, of course, we are talking about maximizing also. Let us start with the first one. I talk about numerous stomata. Numerous stomata. You're going to associate numerous stomata with what? Higher rate of transpiration. Numerous stomata will always be associated with higher rate of transpiration. Very simple to understand. Next is we talk about the rolling of the leaves. Rolling of the leaves. This we will associate with
lower surface area exposed to the sun.
So rate of transpiration will be more or less lesser. So quite a number of times, either we talk about on bright sunny days, there is rolling of the leaves. Bright sunny days to minimize the water loss when the soil is comparatively getting dried out. The plants have to conserve water in them. So we get to see the wilting and the rolling of the leaves happen. Next is, when we are talking about the adaptation, what is the next day? Falling of leaves. Falling of leaves is also there, no? Then we talk about, the cuticle. We'll have to understand all this one by one. Then next is the sunken stomata. Okay, let's try to understand this. When we are talking about the third, falling of leaves, in which type of environmental conditions we generally understand falling of the leaves happening? Dry environment, where in a particular season when the soil is comparatively getting dried out, what happens? Majority of the plants have a tendency of shedding their leaves to minimize the water loss so that the water can be conserved in the stem. So that in the next season, the process of fruiting and flowering can happen. Thick cuticle, we had already been talking about. Thick cuticle in certain plants do not allow the water loss. Sunken stomata. What is the meaning of sunken stomata? Now, in these type of leaves, most of the leaves, what happened there? Good night.
Good morning.
What happened? अबे good night में time है.
Good morning.
हाँ. क्या हो गया? Headache. हाँ. Biology class में headache होता है.
देखो मैम फिर भी बाहर जा रहा.
हाँ. I am talking to you. क्या हुआ? हाँ? Before he opens his mouth, you all are opening your mouth. Why? God has given him mouths to talk. Let him talk. No, I'm asking him one question. If you're not feeling well, you may go out for 5 minutes and come back again. Relax. Now, when we are talking about sunken stomata, what is the meaning of that? Oh my gosh. Too much. The care is too much. Sunken stomata, certain plants, certain leaves like the nerium. Sunken stomata. What is the meaning of sunken stomata? This is what I'm showing you here, where we are talking about quite a number of plants also. If you look into their sections, you will not generally come across, along with the reduced number of the stomata, they will not have the stomata on the surface of the epidermal layer. They may have the stomata in between. Hidden stomata, sunken stomata meaning hidden stomata. So the stomata would be somewhere in between the layers of the leaves. So just imagine, I'm talking about that there's a particular leaf where the stomata are not arranged on the lower epidermis. Rather, the stomata is here. Rather, the stomata is here. Will the water loss be more in such type of leaves? Obviously not, because if the stomata is more concentrated here, easily the water loss. Less stomata on this and more stomata inside, water loss is actually less, no? So when we are talking about sunken stomata, remember one example. So this is the rolling of the leaf that we are talking about. Thick cuticle. Now next is
Can you do the plant area?
plant narium. Narium do I have here now? I don't think so. Bolo. Ventral is upper. Dorsiventral leaf.
And dorsal is behind.
Yeah.
Dorsal is
Behind.
could you explain that
dorsiventral
and isobilateral? Isobilateral, when we are talking about the monocot leaves, isobilateral, where the upper surface and the lower surface is the same.
How do you understand if the leaf is dorsiventral or
Basically, when you talk about also by the venation, you can understand. When you talk about reticulate venation, isobilateral, monocot. And if you talk about parallel venation, that is dorsiventral dicot.
parallel is isobilateral, right?
Parallel is dicot.
No, no, I'm
Sure.
What is given?
I have written that.
Parallel is parallel is mango. Mango is dicot. Parallel is mango. Mango leaf में कहाँ पे तुमको reticulate venation मिलता है? Reticulate venation. People also parallel. Reticulate is this pumpkin, gourd, cucumber. These are reticulate. Karuna. When you talk about check, if I'm wrong, I may be wrong also. But parallel is mango. Mango is parallel venation, and mango is dicot. And pumpkin or gourd, these are reticulate. ये तो reticulate है. And these are monocot. Tulsi, correct.
Dicot has reticulate.
Dicot has
Reticulate.
Yes,
dicot has reticulate.
Yes, ma'am.
Mango is reticulate or this one.
mango को parallel होगा, बाकी सब reticulate हो गए.
Mango is parallel.
People
Mango is parallel.
Reticulate.
Mango is reticulate venation. Show the mango leaf. हाँ तो ये parallel है ना. Mango leaf में ऐसे होता है clean.
हमारा parallel ही है, but our pinnate.
During classes no.
अच्छा
I will check later. देखो parallel is this way, no? Parallel ऐसे होगा. हाँ, this is parallel. Reticulate कैसे होगा? If I talk about, say suppose the rose leaves, हाँ, कैसे होगा? The rose leaves all in the form of a network, no? All in the form of a net. इसको बोलेंगे reticulate, इसको बोलेंगे parallel. And this mango is a dicot. Mango is a dicot, no? Check properly.
People have reticulate variation.
People, कहाँ पे people? It's a clean leaf. People is parallel. Check करो. Isobilateral leaf check करो ना. Dorsiventral leaf check करो. Check dorsiventral leaf. क्या हुआ? Dorsiventral leaf करो. God? Pinnate. किसमें? मैंगो में पैरेलल है. मैंगो में पैरेलल है. Will you be able to remember so many examples that you are talking about? I'll show you where. Rajma dicot. Rajma.
मैंने टाइप किया what type of venation does a mango leaf have? Pinnate reticulate.
No, mango has parallel. No?
I just got good.
So far, if I am not Confused.
Bikes and intensity.
Wait. 1 minute. On the edges of the leaf, based on the shape. Wait, I'll check. No, wait. See, parallel venation is same like the banana and the mangoes, all these, no? Bolo na. Just in 15 minutes you can go. Wait. I'll first of all give you dorsiventral. You want to know dorsiventral leaf. So dorsiventral leaf, just to remember, what I told you is dorsiventral leaf is always for dicot. Dicot ke examples do batao. Dorsiventral is always dicot. Bean, mango, dorsiventral, okay. Tulsi is not dicot. Dicot. But it has seeds, no. So when we are talking about, remember here, just 1 more minute. If you have a doubt between if you have a doubt between dorsiventral and isobilateral, you have to understand by the venation. Now, in your question paper, you have to add it because you will not be asked, you will not be given any clue about the seed. You will be given examples of the leaf. From the diagram of the leaf only, you will have to understand. And if it is reticulate venation, you understand it is a, from the reticulate venation, you have to understand it is a isobilateral. And from, I'm sorry, what I said, isobilateral is monocot and dorsiventral is dicot. So that one question does come on that. So that much only you'll have to remember now. So what I said, we were talking about sunken stomata, and this is what we were looking into. One example of sunken stomata you were asked also in the paper. Nerium, underline that name in the figure in the textbook. There, nerium is an example which has. पैर कुछ ज़्यादा लंबे हो रहे क्या तुम्हारा? Now, next is we had already been talking about the concept of stomatal transpiration. Next is we also, after adaptations, we also have to look into certain factors that affect the rate. What are some of the factors that affect the rate of transpiration? Number one, intensity of sunlight. Here, unlike the chapter on photosynthesis where you had studied a detailed process about how light intensity and carbon dioxide affect the process, here you have to only understand whether the rate of the process and the factor increasing, decreasing is directly or inversely proportional. Only this much here for transpiration. The detailed process is not necessary for you. Intensity of sunlight is how the rate of transpiration will increase or decrease. So directly proportional. Remember, with increase in the. What an explanation. So now, when we are talking about intensity of sunlight, there. One characteristic common to all the tanmais is they talk a lot. Unnecessary वो भी. ये 9th standard में थोड़ा सा सुधरा हुआ था, अभी तुम्हारे साथ आके तो और भी बिगड़ गया. तुम तो बचपन से बिगड़े हो. I hope, I feel. That is what I can comment in this few months. Intensity of sunlight. With the there. Oh my God, what a class is this? अब क्या है? What was this? किसका था?
Water का point है.
Notes clear.
अभी मैं तुमको निकालू क्या क्लास से? Go and sit at the last bench. Get up immediately. I don't care. Get up and go at the last bench. If you have to sit in the first few benches, you have to behave yourself very well. You have to be a well-behaved person. Fatafat, take up your books and go at the last bench before I will waste another minute. Fast. Intensity of sunlight. Now we talk about temperature. With the increase in temperature, the rate of transpiration should increase, because again, temperature has something to do with the opening up of the stomata. Velocity of the wind. Directly proportional. More the velocity. Why but? More the velocity of the wind, why the rate of transpiration is more? Because the surrounding of the leaf is getting dry. If it is windy, is it possible that the surrounding of the leaf gathers adequate amount of moisture? No, because the wind is driving away the moisture. The wind is driving away the moisture. So this process that just now we studied can be easily carried out. But if it is not a windy day, but at the same time a monsoon day, what will happen?
More moisture will be there.
More moisture will be here, then the release of moisture will be comparatively less. So the amount of moisture will get stuck up in the leaf intercellular spaces, and the uptake of the water will be comparatively less now, because the leaf is already having so much of water. So now we are talking about humidity. Just now I said humidity. More the humidity surrounding the leaf, lesser will be the rate of transpiration. Carbon dioxide concentration. Higher the carbon dioxide concentration, lesser will be the rate of transpiration. Why? The reason is higher the carbon dioxide concentration. Now, what is the average amount of carbon dioxide concentration that can be tolerated by the guard cells? 0.03 to 0.05. Minimum is 0. 0.03 minimum. Optimum is 0.05. Utna tak the guard cells can tolerate. But above 0.05, as I said, in case of photosynthesis also, above 0.05, it is considered as a pollutant. Then, with higher concentration of carbon dioxide, why does the rate of transpiration drop? Because of the closure of the stomata. At a higher rate of carbon dioxide, higher concentration of carbon dioxide surrounding the leaf, the stomata starts closing down because of the inactivation of a variety of enzymes. Kirtana. Girls behave like this in the class. Listening to you. Now, when we are there, I really want to meet your parents now. So atmospheric pressure. Now, when we are talking about atmospheric pressure, higher the atmospheric pressure, lower is the rate of transpiration. No, if the atmospheric pressure is more, again, the process of diffusion is getting disturbed. Water content of the leaves, what do you think? Iron. Water content of the leaves and the rate of transpiration, what should be the relation? इतना सोचना पड़ रहा है ट्रांसपिरेशन के लिए. More the water content, higher would be the rate of transpiration. So this is what we are talking about. Adaptations already we had been looking into. Now specialized or modified. Now here we talk about some specialized modifications of the leaves. Leaves converted into spines or needle-shaped leaves. Especially we talk about the xerophytic areas, no? Where we understand that in the dry areas where the soil is constantly dry. What type of leaves we basically come across? Needle-shaped or leaves which are modified to form spines. Why? To conserve the water, minimize the water loss. So this is, yes. Surface area, correct, to reduce the surface area. So these are some examples that you'll have to understand. Experiments, I don't think we have any more in this chapter. Now, take up some questions fast. Let us try to understand some of this context. Now, there is one more definition that we have to understand with reference to transpiration. Now that's a major difference that you also are asked in your paper. When we talk about transpiration, we have been talking about transpiration, and now we also try to understand one more word here known as guttation. What is guttation? Did we study guttation earlier? Absorption mein padha tha na? So now you tell me two features related to this, or two conditions, characteristics related to guttation. Aishwarya, number one. Guttation ke saath related something, you tell me. Anything related to guttation that you remember? Hydrothos, correct. Hydrothos. What else?
Droplets of water.
What else? Droplets, okay. Same thing. Next, droplets. What else? Prisha.
Actually,
ये इसको nickname है exudation.
guttation.
It happens first in the morning or later.
Okay, so when we are talking about, especially when the rate of transpiration is not carried out. So as it says, okay, early mornings. So what are we trying to understand for guttation is a particular process which would generally happen when the water accumulation is too much in the plant, when the rate of transpiration cannot happen because the stomata is closed. So at that time, from where does this happen? From which surface of the leaf does this happen? When I talk about hydrothos, where are hydrothos present? On the leaf margins, and present on the leaf margins. And stomata always we understand from the lower surface of the leaf. No. So that's a difference. Can you now tell me three comparative differences between these two processes, guttation and transpiration? The first difference, guttation means loss of water in the form of droplets, and transpiration in the form of vapor. Second difference,
Of the release of water vapor. It is from stomata and
Lenticel and
cuticles, and in guttation it is from margin.
margins of the leaf, and when, from the ending of the vein, there's a specialized pore, and that is called. So the second difference always you should write, transpiration is carried out by stomata majorly. Don't compare between the hydrothos and lenticels. Say between the stomata, and the stomata majorly contributes. No, stomata and hydrothos. Third difference between transpiration and guttation, I'm asking you. Third difference. Third difference.
And transpiration of the dry, yes. And
Guttation. Not necessary. Guttation in when you are talking about winter mornings, also you get adequate amount of water in the form of droplets clinging to the tips and the margins. What is written there?
It's written. It occurs during dry conditions, and guttation occurs in humid conditions.
Transpiration occurs in dry conditions, meaning when the wind is hot and dry. High rate of transpiration. Dry meaning we are not talking about the soil dry. We are talking about dry condition where the air is hot and dry. The atmospheric conditions are hot and dry. But guttation you will come across now also in the monsoon time. Will you come across guttation?
Yes.
Monsoon time will you come across guttation? Yes. Early morning you will come across, especially if you have those potted plants in your windowsill. You check. Early morning you will still come across small droplets on the tip of the leaves. Will you come across this process in the summer? Summer is hot and dry. Will you come across it? Yes. Early morning, you check. Still, you will come across, because all through the night the stomata was closed. All through the night the stomata is closed, but provided the solace having adequate amount of water, then only guttation will happen. Winter mornings, will you come across? Yes, very much. Haan, bolo, fatapat.
Now in transpiration, water vapor is lost, but in guttation, other fluids are also.
Correct. That's a very important point to remember. When you're talking about guttation, it is the loss of the sap, water. So along with water, there are so many essential minerals the water, the plant loses. But for transpiration, it is in the form of vapor. So the mineral loss is not there.
Transpiration has a cooling effect on the plant and guttation.
No. Now we talk about something else.
No.
But no. Yes. Yes. But now when we are talking about guttation, and there is one more word that we have to talk about, bleeding. This is also you get in your paper. Bleeding. Now bleeding with red is also. Bleeding. When we are talking about bleeding, what is it? Is it the same like guttation?
No.
What happens?
So when cut.
When we are cutting or injuring the plant part, then the release of the sap and the minerals and the salts. So the release of the substance is same like guttation, but the process is not the same. For bleeding, we are not talking about any specialized surface or a region or a pore or an opening. No. So
It can take this and
excellent. So now let us talk about some questions. We'll do some questions, and I think we should be done with this chapter. I want which of the questions that you should try. Give me a pencil, please. Now, B number one. B number one. Name the following. I'm going to discuss as much as possible. B number one. Name the following. Start with it. B number B.
It's done already on the subject diagram.
In the class.
Yes.
In
We
which
can do
school?
it from this.
Wait. From the textbook.
Everyone has not done.
Yeah, we can do it from.
No, and we can. It's not done.
If you allow me to talk. Which of the questions you are supposed to do it, of course. I'm not asking you what you have done in school. B sub number 1 you have to do. Check B sub number 1. C sub number 2. Sub number 1 also. This type of questions also you get. Sub number 3.
See no.
D sub number 2 we just now discussed. D sub number 2 we discussed already. And then E sub number 1. Now all these diagram based questions you should be done with and clear with. Number E sub number 1 करना है 2, 3, 5, 6, 7. Most important is number 1. These are as it is questions you get in your paper. As it is questions you will get in your paper. Number 1, number 3, number 5, 6. These 4 questions are very, very important. I'll give you 5 minutes. Those of you have not done at least this diagram based questions, I'll ask you to go through. Doubt nahi hai, then I'll go to the volume questions. Volume agar hai, those of you have not finished with this diagram based question, first we do this. Go through. And tell me if you are sure with all the answers. First of all, these 4 questions of the textbook. Those of you have done with this, take out your volume. Otherwise, all of you, first you take the questions. Previous board questions, volume. है सबके पास?
Yes.
नहीं है.
Yes,
So
ma'am.
let's write it down. अब you have to solve at home. Take it at home. Page 22, 72. Page 72. Page 72. See the same question. Number 5. Previous board questions only I'm talking about. 75. Page 75. Number 5. No, 72. 72.
72 पे करना क्या?
Number 5 करना है. 72 I said.
Number 5.
72 का number 5. 72 का number 5. Number 7. Number 7. See there. Same questions. Number 8 also the same. Number 9. Check करो. Number 9. Number 10. Number 12. Number 12. All these are the same questions. Force of gravity is so much. Or your maturity is less, I don't know. 21. We have just now discussed about 21 and 22. Chalo, at least those of you who been into the questions of the text, please start doing the questions of the volume. And jinka page 66 or 67 के questions नहीं हुआ है, first we will solve that. Text के questions पहले करो. Diagram based only. The rest objective you can try at home as homework. Karo, fast. All done? This diagram based questions all done? Okay.
And next class we will explain.
Garrow's photometer.
These are complicated, they won't come, right?
These are actually not there in the syllabus, but in your school exam only you will get.
Explanation, but it's
What is there? What?
and then we think
The vowels and suffix. What is here? You are taking up upper vowel. These are two vowels. Between the two vowels, you place a leaf. What did you take here? Calcium chloride. Calcium chloride. Why do we take calcium chloride? Calcium chloride can absorb moisture. Calcium chloride can absorb moisture. Now, what are we doing? We are placing this apparatus under the sunlight. Oh, let's start with those questions. Any doubt? You ask. Next question, पहले हो चुका है. Then you move on to the following questions. Then now what? Wait for a particular period of time. Now you have to understand that in this upper barrel you are fixing a bent glass tube. What are you taking in this bent glass tube? Mercury. What are you taking in this bent glass tube? Mercury. In this particular period of time, when you are working under the sunlight, what are you supposed to see? You are supposed to see that in this wine where there is calcium chloride, the release of the moisture. Forget about calcium chloride. In which surface of the leaf you are expecting a higher release of water vapor? Lower surface. So you are understanding that there will be a release of water vapor more in this surface. Upper surface comparatively it should be less. And then when you are talking about the calcium chloride will also start absorbing the water vapor. Whatever amount of water vapor is there, calcium chloride will absorb. Then in this case, what's the purpose of it? Okay. After few hours, calcium chloride is taken. After few hours, what is mentioned here? Equal amount of calcium chloride is taken. Placed into it. On the ends of the floor, fine. Taken out. After few hours, the calcium chloride vials were taken out. We are not to actually only measure the amount of the mercury going out, but we are also to measure. We are actually to measure the calcium chloride vials. So now that we talk about the calcium chloride vials, we take out. Which should be giving you higher grade? This one. Why? Loss of water vapor is small. So this will absorb more water vapor. So this will have a more amount of the dissolved form of the calcium. So now in this case, since it is having so much of water vapor, yeah, that is what we have to come across. Automated design groups. But in there's no solution in that. Major this automated is the calcium divide. Also, we can see the units of the market. We can see the complete these questions, okay, all of you.
ऐसे बैलेंस नहीं करते ना मैं
तुम लोग यहाँ पे तू जा रहे तो जल्दी जाने
कोई ऐसे
इधर बैठना
इसके पास
मेरा भी